Hello, everybody. Today we're going to talk about meiosis. When we did mitosis notes last week, some people were a little bit confused, so I thought I would go through these so you can kind of understand the process of meiosis. In many ways, it will also explain the process of mitosis as well, since they are related processes. The only difference is mitosis is trying to create a cell that's identical to the original cell. Meiosis, we're trying to create um, sex cells. So they're going to have some differences there, and that's what we need to look at. All right, so as you can see, as you go through the process, the phases are still named the same. We're basically just going through it twice because the goal is to not only reduce the amount of DNA in half, that was mitosis's job, basically created double the DNA and then you divide it in half and you had two cells with the same amount of DNA that you started with. Here we're trying to do that and reduce the total number of chromosomes. We need that or we're going to have some issues. So if you have too many chromosomes, you will have some sort of birth defect. So to prevent that, meiosis reduces the number of chromosomes in half. So your sperm will have half the amount of DNA that your regular cells have if you're male, and if you're female, your eggs will have half the number of DNA as well. There is some benefit to that. We get some genetic variation, so you're just not giving your parents' chromosomes, you're giving a mixture of your parents' chromosomes. And that's combining with a mixture of your spouse's parents' chromosomes. The only drawback is finding a mate, sexual reproduction, you need two. As I said, the phases are very similar. We start out with interphase, and we're going to go through like one cell division, which we end up with prophase. Prophase has some little differences in it, though, that we're going to talk about a little bit further. It's called crossing over. So you can kind of see the chromosomes here are combining with each other to form what's called a tetrad. So instead of just two chromatids, there's actually four ended up here. One's from mom, one's from dad. They're going to come together there and do some crossing over in between. So that's going to create some variation. I'll go into crossing over in just a minute. But notice in metaphase, it's not just one line of chromosomes. You got a double line where the number one from dad is combined with the number one from mom. Number one from mom, number two from mom, number two from dad are there. They can be in any order. That's another way we get some variation. These things can flip-flop. Anaphase. Notice we're not pulling apart the chromosomes. We're pulling full chromosomes to each side. So this does end up reducing the number of chromosomes. So before we would have, these would be lined up and we'd have four chromosomes being put over in mitosis. Here we only have two chromosomes. So we're already going to be reducing the number of chromosomes in half. We just got twice the amount of DNA we need. Telophase is pretty much just like telophase was before. You're going to make that nuclear membrane again. Cleavage is going to start, and we're going to do cytokinesis and create two new cells. So these cells now are what are called 2N. They got half the number of chromosomes in each cell. They just got twice the amount of DNA. Now, it might seem silly that we make nuclear membranes and then go into meiosis 2, and the first thing we're going to do is get rid of those nuclear membranes. But you got to remember this is evolved from mitosis, so a lot of the same procedures still are needed here. Now in prophase 2, again, the nuclear membranes, this is going to look just like mitosis. So we just got half the number of chromosomes. So the chromosomes line up in the middle in metaphase 2 and anaphase. Those chromosomes are pulled apart to chromatids, and so that you end up with one chromatid from each type on either side. So we'll end up with 2, 2. We've got half the number of chromosomes and half the amount of DNA like we're supposed to have. And then we'll go through telophase again, dividing the cells again in half. And if these were sperm, you would have 4 equally sized cells that would then mature and become sperm. In females, the eggs will divide a little differently. They'll end up with just one really, really large egg cell, and the other 3 will be smaller uh, pieces of cells that'll kind of be discarded. Only the one large cell will be. All right. So let's look at chromosome number and what does haploid and diploid mean? Well, diploid, if you recall, means that you've got 2 sets of chromosomes. One set comes from mom and one set comes from dad. When we become haploid, we get rid of one of those sets. We only end up with 1 set. So we got half the amount of DNA, so that when we combine with our spouse's DNA, we'll have the correct diploid number. If 2 diploid cells would connect, we would have way too much DNA and it wouldn't work. So if we started out with 3 chromosomes, that's the 1 end. So in humans, that would be 23, because we have a normal cells, you got a total of 46 chromosomes. So our sperm and eggs are going to have 23. So that those 2 sperm and eggs combine, 1 from each parent, and then we'll end up with the right number, 46 in humans. All right, so I did mention that in prophase 1 of meiosis 1, so the very first stage, there's some differences here. The chromosomes are going to line up with each other. So you got that double line of chromosomes. In a cell, 1 chromosome is going to come from the male, which in this case is the yellow one. 1 comes from the female, and that's the mother. So it's been duplicated. So we end up with 2 chromosomes that have been duplicated. So we got double the DNA at that point. Genetic information is the same. On this green one would be the same as on this green one. The DNA on this yellow one would be the same as on this yellow one. They are the same. Their size is the same. Their position's the same. The banding pattern's the same. They're all going to be identical. And those are referred to as homologous chromosomes. Homo meaning the same. They're the same chromosomes. We're going to start out with those in prophase. So they're going to come together. One from mom and one from dad. In meiosis 1, which is the reduction division, where we're going from DNA 2N to the haploid state, we're going to create all sorts of genetic variation at that point through the process of crossing over, and then the homologous chromosomes are going to pair up and separate, and that's going to create variation. So not chromatids, so we're going to have the full chromosome. So full X-shaped chromosomes are going to get pulled over to each side. One thing that's different. Now, what is crossing over? Basically, it's exchanging genetic material between the 2 homologous chromosomes. This gives genetic variation because the chromosomes will end up not being identical. So if we have here the father's chromosome here and the homologous chromosome of the mother's here, meaning they got the same genes on there, it's just they might have different alleles. They're going to basically cross arms, and where they're touching, and basically switch arms, so that you end up a piece of the mother's here and a piece of the father's here. And this is going to occur on both sides, on different parts of the chromosome. So it's going to end up with 4 distinctly different looking chromatids when we're all said and done. All right, and then in meiosis 2, this is going to look just like mitosis. There's no interphase 2. We've already duplicated the chromosomes. We don't want any more DNA. We're just going to split it up to the normal amount that's supposed to be there. And then we create our product. So we're going to end up with 4 new cells. They are genetically different. They're going to have half the number of chromosomes as the original. So just to see how they're different. So mitosis and meiosis. So this one, mitosis for asexual reproduction. Meiosis is sexual reproduction. So meiosis had one division. You only have one parent. You end up with two daughter cells. And the cells are identical with the same number of chromosomes. And basically, they're going to be the exact copy of each other. In meiosis, we're going to have two divisions of the cells. So we're going to need two parents to create a child. Each cell will end up with four daughter cells as opposed to two over here. The cells will look different than the original parent cell that created them because they got different genes on their chromatids. They got half the number of chromosomes, so they're considered to be haploid. This is only done in your gametes, so either your sperm or your eggs, and it creates a lot of genetic variation if that bar goes away. There you can see that genetic variation is created with meiosis. All right, when we get to genetics, we'll come up with meiosis again, so that's coming up pretty soon here. And when we talk about the laws of heredity, so I'm just going to kind of skip through this pretty quick, so we'll talk about this more in genetics. When we looked at Mendels Laws of genetics, he came up with these laws of heredity. They're basically explained through the process of meiosis. So keep in mind, don't forget meiosis. It's going to come up again when we talk about genetics. Both of these laws are very important. All right, so that ends our notes on meiosis. If you have any questions, make sure you email me with any problems you might have, things you don't understand, and so we can straighten all that out. All right, thank you for listening. At this point, you should be ready to do the online quiz. So it's just a Google form. Answer and make sure you submit it. That's it. Goodbye.